Guys, this is just a quick overview recap of our Unit 5, which is covering photosynthesis right now and then cellular respiration later. So let's go ahead and kind of break down where we're at so far. So as mentioned in class, where we're at as far as photosynthesis goes, we are looking as though we are zooming in on our leaf. So if we were to take a section of that and zoom it in, and we would have all sorts, we'd have all of our plant cells kind of looking like this. And then inside each of those cells, and this entire area would be full of that, but inside those cells, remember that we have those little chloroplasts. So if we were to zoom in on one of those chloroplasts, we would see something that kind of looks like this. And then inside of the chloroplasts, let me zoom in a little bit here. Inside each of those chloroplasts, remember that we would have these stacks of discs, and each of those discs, each of those little green lines, each of those is going to be a thylakoid. So a single thylakoid, but when we have a group of them together, that is called a grana, or a singular would be granum. And then filling the rest of this space would be this more like light green substance, which is what we call stroma. Okay, so that's what we're looking at so far. And if we, again, kind of recap, as far as, come back. As far as photosynthesis and cellular respiration goes, our light-dependent reactions, those are going to be happening, again, inside of the thylakoid. So let's make that teeny tiny here. So our light-dependent reaction is going to be taking place inside the thylakoid, and then our light-independent reaction that's going to be taking place in this lime green area of the stroma. All right, so that's where we're at so far. So if we were to take our chloroplast and kind of make it a little bit bigger, which I'm going to do here in just a second, we're going to be able to see all of our products and all of our reactants and how things get ripped apart and rearranged. That's that part of like carbon fixation. And then we're also going to see what happens with ATP turning into ADP pi, and then also NADPH turning into NADP plus. So we're going to take a look at all of those things. But again, that's just kind of where we're at right now. So we're inside the leaf. If we could blow up that little section of leaf, we would see all of the little teeny tiny plant cells with the little chloroplasts inside of them. And then this is what we're looking at for each of the chloroplasts. All right. So let's go ahead and get into what this looks like within the chloroplast. Okay. There. Okay. So in our drawing of the chloroplast, which kind of looks like this, there we go. And again, we're going to have that thylakoid stacks kind of looking like this. And now each of these thylakoids is going to be doing the light-dependent reaction. So each of these three discs right here, each of them is going to be doing the photosynthesis. And inside of the whole chloroplast, because remember we're in the chloroplast right now, you're going to have multiple grana. So you're not just going to have this one singular stack, you're going to have multiple stacks all throughout the whole thing in a three-dimensional shape. But for right now, we're going to keep it somewhat simple with just one stack, and then we're going to fill in the rest of our chloroplast with this lime green, which is representing our stroma. Okay. And then right down here, we are going to also write our chemical formula for photosynthesis. So we're going to take 6, I want to do this in black. All right, so we're going to take 6 CO2 molecules plus 6 H2O molecules will give us 1 molecule of glucose plus 6 molecules of oxygen gas. So we're going to have this in black for right now. As we go through it, we're going to, again, kind of see where all of this stuff is coming and where it's going. Oh, we also need to include light energy as kind of a catalyst. Okay, so we'll come back down to this and we'll refer back to it as we go through. Okay, so to start this process off, we're going to take a look at how we're going to take a look at the beginning. So first thing that's going to happen is light energy is going to come into the thylakoid. And as that light energy is coming in, remember that it is going to essentially make it so that water, and I'm going to draw water in kind of an interesting way. I am going to do water coming in, and that's going to be H2, and then we're going to make the oxygen in blue. There's going to be a method for this. There's method to my madness here. Okay, so H2O, so water is going to come into the thylakoid. Okay, so then down here, we're going to give it the same colors. Okay, so we're going to do H2 and then O. All right, so the water comes in. It essentially gets electrocuted inside of photosystem 2, and we'll draw that part and kind of outline the major parts of the light-dependent reaction. We'll take a look at that in just a minute. And so as that's happening, because the H2O is getting ripped apart, okay, so instead of it being one molecule, we're going to rip the oxygen away, we're going to rip the hydrogens away. And so as that happens, this oxygen molecule is now going to produce oxygen gas as a waste product. Now, that will become useful later when it comes to cellular respiration. So this blue oxygen is going to be the source for our oxygen gas as part of the products. Okay. Now, our hydrogens. As the hydrogens are getting ripped apart from the oxygen, it is going to release electrons, and those electrons then get energized by the chlorophyll, or with help from the chlorophyll, and they get the energy from the light. Okay. Those hydrogens are going to make that hydrogen gradient because we're going to be on the inside of the thylakoids, and we want to make our way to the outside. We want to make our way out to the stroma. So as the hydrogen is building up that hydrogen gradient, it needs to go through an escape. And in order to do that, it will go through, it'll do two things. The hydrogen will go through ATP synthase, which is our protein that puts together adenosine diphosphate plus that third phosphate group to make adenosine triphosphate. So the ATP synthase does that. And then the hydrogen is actually going to hip to ride with another molecule, let's make it orange, which is NADP plus. So NADP plus the hydrogen that came from water, here's our hydrogen from water, that's going to take a ride over to the Calvin cycle or the light independent reaction. Now, there's another molecule that's produced that's also going to hitch a ride over to the light dependent reaction, and that's going to be ATP, adenosine triphosphate. Okay, so it's going to hitch a ride over into the stroma to help fuel the Calvin cycle. Okay, so that's our overview of the light dependent reaction. And again, we'll draw that out more of like what I did with the animation. I'll draw that out again. Okay, so now we're going to come over here to the Calvin cycle. Remember, we're in the stroma of the chloroplast. We have now got out, so we've left the thylakoid. We're outside of the thylakoid. We're beyond the membrane. Now we're over here in the Calvin cycle. So I'm going to draw my Calvin cycle just like this. So remember, there's 3 parts. We don't really need to know the names of the 3 parts. There's 3 parts to the cycle. And so now in the Calvin cycle, yes, we're getting NADPH, yes, we're getting ATP, but these are just like helper molecules. So we need to also now include our second reactant, which is CO2. All right, so let's go ahead and add some CO2, and I'm going to change this color to something a little bit easier to see. Let's go purple. Okay. All right, so now we're going to get C, and then let's change the oxygen to, change that to a red. Okay, and then O2. So we come down here, we're going to come to our CO2 molecule. Here is our purple C and our two red oxygens. Okay, so CO2 is now going to come into the system. Again, also it's coming from the atmosphere. This is just naturally in the atmosphere. And I do also want to mention oxygen gets released into the atmosphere. It looks like it's being released into the chloroplast, but it'll either get released into the atmosphere or it will also make its way over to the mitochondria. Different discussion for a different time, but just know this oxygen gas doesn't just hang out in the chlorophyll. All right, so the CO2 comes in. As it's coming in, this ATP molecule, so we're going to move, oh, I just want to move my ATP molecule. Our ATP molecule from right here, it is going to be used as like our fuel. Okay, so ATP from this molecule here is coming over into the system and it's going to be used up, and then that's when it turns into ADP plus a phosphate group. We also see our NADPH. So NADPH is also going to come over. And that's where that one's coming from. So it's coming from this part right here. So the NADPH, NADP, and then we have our bright pink H. It's going to be used up where it will now turn back into NADP plus. But this little hydrogen, this little guy, it's going to be used with the CO2 to make our glucose molecule. So we're going to give off, so this is coming off of the Calvin cycle, and that's going to be giving off our purple C. So that's our C6, H12. So see how this is, our H12 here is pink? That's because it came from our water molecule. Hitched a ride over with NADPH, and now here it is in the sugar. And then we're going to also give a red O2, or excuse me, O6. Okay? Because this red O6 came from the oxygen in CO2. So we can do that again. So C6, purple carbon, so from the CO2. H12, I keep messing up my colors here. It's our pink H12. Oops, that's supposed to be the same color because it hitched a ride over from the water. So pink H2, hitched a ride on NADPH, got ripped apart again. It's going through a lot. Turned into H12, pink H12 from the water. And then our red O6 came from the CO2. Now, our cycle's not quite done yet. See how we're at the bottom of the cycle? So we actually need to use up another ATP molecule. We actually use 3 more here. To get the cycle back up to its original state. Actually, let me get rid of that. I'm going to get rid of that I. There we go. Okay. So this one uses 3 ATP. This one uses 6 ATP to get there. Okay. So now we have these kind of molecules hanging out that aren't really doing anything right now. So they're used up, they're empty. They aren't doing anything over here in the Calvin cycle anymore. So what we're going to do is we're going to send these molecules back over to the light dependent reaction. So there's our NADP plus, ready to go back over to the light dependent reaction to pick up more hydrogens and electrons. Remember, those electrons are going to be important too to help put together the sugar. And then our ADP, adenosine diphosphate, and the phosphate that got ripped off of it. See how they're separated from each other? They're going to head over to the light-dependent reaction as well to get pieced back together. Okay, so that summarizes again kind of the whole process in an overview. So let's take a look at the electron transport chain a little bit more thoroughly. Okay, so this is as though we're zooming in on the thylakoid. Okay, and we're going to be looking at just basically the separation barrier that would be found. This would be the separation barrier. Let me try to find a different color that I haven't used yet. Use this one. This would be a separation barrier that would be found right here. It's between the inside of the thylakoid and the stroma. Okay, so let's go ahead and draw that out. So this would look like, and I'm putting it in that dark green color because this is the membrane for the thylakoid. Phospholipid bilayer. Then we'll use this kind of tealy color to make our photosystems. So this is going to be photosystem 2. And then later down the line, we'll put photosystem 1. And then a little bit further down the line, we will go ahead, and I'm going to use that same color that we used for ATP up above because we're going to be creating our ATP synthase. So this is ATP synthase. All right. Now, in between photosystem 2 and photosystem 1, there are going to be 3, let's make them, I'm going to do 3 blue blobs. All right, and these 3 blue blobs, bloop, bloop, bloop. These are part of the electron transport chain, and then we're going to actually get a couple more blobs here. Bloop, bloop. Okay. And then on the bottom here, this would be the inside of the thylakoid, and then up here where all of our formula is, this would be the stroma. This is the outside of the thylakoid. So again, we have light energy coming in, and that light energy is going to strike the photosystem 2. We have water, our pink hydrogen and our blue oxygen. Pink hydrogen and our light blue oxygen. That's going to start the process of photolysis. All right. So photolysis is basically when the hydrogens and oxygen, so the water, it hits the photosystem and it kind of gets electrocuted. And in doing so, it rips apart the hydrogen and the oxygen. That's how oxygen can escape to go make oxygen gas. And then the pink hydrogens, they actually come over here. And that will create what is our hydrogen gradient. Okay, so it rips it apart. The oxygen goes away. It's now a gas. It's oxygen gas. And it makes our hydrogen gradient. Okay, and we'll talk about that more in a second. Inside of the photosystem, that's where we would find our chlorophyll. So we're going to make that kind of this greenish yellow color. And that chlorophyll is what helps to excite and move along the electrons. And these electrons are, let's do kind of this grayish color. The electrons are coming from the splitting of water, because remember, electrons are used to help hold together bonds in our chemical formulas, our chemical molecules. And so when the water gets ripped apart, those electrons kind of just escape from the molecule, but they're getting energized. They're getting all of this energy from the light, which goes into the chlorophyll, which goes into the electrons, which then allows for the electron. Actually, I'm going to draw one more so I can move it. There we go. And that's going to allow for this electron right here, and this would be true for all of them. They're going to get excited. They're going to move down these proteins, so down the electron transport chain, where they get to photosystem 1. There's some more chlorophyll in photosystem 1. And then they're going to get excited. They get re-excited in photosystem 1, and then they move to the rest of the electron transport chain. So we went up We came down, we ran out of energy, we had to get re-energized, and now here's where our electron is. Okay, so now our final part is the hydrogens here. They want to be able to move because we've got a lot of hydrogens. It's just a little too crowded. I'm going to draw another one. That way I can move it. Okay, so it's too crowded. We need to move our hydrogens through ATP synthase because they can't just make their way through the membrane because we have to pretend like there's a bunch of these little guys. We have to pretend that there's a lot of phospholipid bilayer going on here. So they can't just make their way through. So the hydrogen, what it will do is it will kind of slink its way through the ATP synthase. And in doing so, it drives the ATP synthase to put together the ADP pi into ATP. Okay. So that ADP plus the phosphate will go through the ATP synthase and makes the ATP. And then just like we had up here, that will now go over to the Calvin cycle. That hydrogen will also make it, so it's made its way through ATP synthase, which helped drive this formula forward. And then it will come over here where it will hitch a ride, or it will work with, it'll work together with NADP plus, and then this electron, there's actually two of them that would be involved here. Let's draw another one. The second electron, because we need two of them. Those electrons would also go with NADPH over to the Calvin cycle. Okay. All right, so there it is. That is one half of our unit in just a quick overview, a quick nutshell. Hopefully you find this helpful with the visuals and the colors. If you have any questions, please let me know. Otherwise, happy studying, guys.